My microphone because the webinar started. So good. It's good. And if I'm able to hold target. Don't mind me if I'm smelling.
So smelly.
I've got sweats right now.
Oh, no. Hello. How are you?
Hello.
Hey, baby.
Hey.
Good to see you.
Good to see you too.
The most popular person at IB is here.
Right now, the most popular person at B is Lucas.
That's what I meant, but
no one
was, did you think I was talking about? Oh,
Exactly.
okay.
But no one will sit around.
You're supposed to sit here so I can throw stuff at you double. So, also, we didn't wear anything for Lucas, like a bandana. Should have thought of them.
We'll be switching over here.
Thank you.
Thanks.
It'll probably take us about 20 minutes just so that I can.
I need.
Yeah, it's. Is there a ones?
Yes, they're there.
Okay. Yeah, please, sir.
How's it going, David?
Okay. It's not just the student that gets nervous.
We've heard him a million times. He's amazing. But you know that already. Yeah.
Cool.
So, thank you, Joe. I'll be right.
Excuse me. Oh, you're back.
Hey, thank you for covering me.
Oh, I
You're
got it, no
amazing.
problem.
I
Yeah.
do want to fucking kill you also. That was so cool.
Tell you what happened?
Can't believe it. I texted Carmen and I emailed her with Russell and I was like, hey, Russell will be covering me. I totally forgot yesterday morning while I was in San Francisco. And Russell emails both of us and goes, hey, or would you say?
That was today?
That was
Exclamation point, question mark, exclamation point, question mark, which I thought might give it away.
the way that I raised out of bed.
I knew you were mad because the first thing you texted me was, dude. Oh, I did not mess around.
Lose my mind.
I can't make
So, that's so. Oh, I was gonna leave, but it's good to. Really? Wait, what was your last email?
One that he's actually just.
Biology.
Oh, with your TA, I'm very disappointed in you.
My heart breaks. Heart breaks.
Because he knows I would have just absolutely
I would have been murdered.
murdered him.
I wouldn't make it in any classes actually.
Worse, I would have just told Lauren he had to be.
Yeah, and I'd be super dead.
Blanket.
Oh, the number.
You should read the books better.
I just rubbed my leg. Yeah. I know, it's like.
Makes it Mars fitting. Maybe I'll.
I know, I didn't think about it.
Oh, I love that.
Thank you. Fine.
It also looks like.
No one's in there until you get there.
I just don't know what direction I want to head right now. House for you, like, dip my toe.
House for you.
See good. For you, no one.
All excited.
Should have been like, nah. I'm saving that for my buddy. Put your jacket over.
Yeah, like I'm like thinking the bass tech guitar. Just like.
Yeah, too much. The whole thing. You just hear
Something I really want.
Yeah, exactly.
No, now you can.
You're talking. You've seen more than, you've seen manatees. A lot of things happen to manatees.
Sure have. Sure have.
But now you're free for 2 years. That's sorry.
Martin's about to steal a seat. How are you? Okay, nice.
Tsunami hits right now.
Hey, nice.
Good.
Pretty good.
I meant to like.
Yeah.
They're just like. I don't know, and then they like wanted to put the earbuds, they plotted for the hammer, and it's just like. It was like. I didn't think.
Oh, nice.
Let me slide through.
So it wasn't too bad.
Welcome, everyone. To the first part of Lucas Morimoto's defense of his masters. So Lucas contacted me in November 2019 about working in the lab. And I get contacted by students, undergrad students, and Lucas stood out in that he included in his original email to me examples of some of his artwork. And so that like caught my eye in ways that a student typically doesn't catch my eye. And although you don't have any of your artwork being shown here. Oh, good. Oh, excellent. Good. I was thinking I was remiss in not having some to begin with. So he officially began work in my lab as an undergrad on the 10th of March, 2020. Now, if you remember exact dates, it was later that week that everything shut down. So it wasn't, that complicated things rather dramatically in terms of what research he'd be doing in my lab. And because of the remoteness that that was enforced, we ended up transitioning so that his project in the lab was to do a graphic novel telling the story of one of the research projects in the lab. And this is what I should have shown here. Some of the pages from that. They're absolutely incredible. They're beautiful. And that went on for quite a while. And then we got back into the lab and things were functional. Lucas managed to live through 3 lab moves during that time. So my lab moved 3 times. And we eventually transitioned back here, of course. Lucas began in the fall of 2022, while we were still out at the Post Range building, to be a master's student in my lab. Because of his interest in genetics, we chose a project that was looked to explore genome-based methods for species delimitation within some of the beetles that we worked on in the lab. And the project that we chose was one that explored further a project that John Sproul had looked into, having to do with repetitive DNA elements and how they could be used for species delimitation. And so from that starting point, Lucas's work, as is often the case in graduate programs, grew into various other aspects that we hadn't imagined. And that's what Lucas will report on. One of the things that I wanted to say that during Lucas's journey, he has blossomed in many regards. One of the ones that you might not know is that he's quite good at guessing potato chip flavors now. But more importantly, his project became more focused on the analysis of genomic data rather than the generation. I mean, if he generated the data, but he became more and more interested in the bioinformatic tools and has become quite adept at that. And I think that that's one of the things that I'm most proud of is how you've grown in that way. These are transition times that are always difficult for me. These students grow up in your lab and, they're like your offspring. And so we will miss you, Lucas, wherever your future takes you. But we will be happy to hear what you've been doing the last few years. So.
Right. Thank you, David, for that introduction. Yeah, so my name is Lucas. Thank you all for coming here today. I'll be presenting on my master's research that I've been working on for the past several years. And I've decided to title it Lines in the Gravel, Comparative Evaluations of Standard and Novel Species Delimitation Methods in Cryptic Bembidian Species Groups. So a bit of an overview of the presentation. I'm going to start off by asking what is a species, or rather how I will be considering it for this study. What is species delimitation, some standard and novel methods, getting into the actual study system that I'm working with, the genus Bembidian, some experimental design, methods, aims, results, and then finishing off with conclusions. So, get it out of the way, what is a species? It is a rather contentious question, and it has been for a number of decades. There's kind of an inherent conflict between biologists' desire to have a practical and replicable definition that they can use, along with sort of the cryptic nature of evolution across the entirety of life as we know it. There's always sort of an exception to a rule. And so because of this, as of 1997, there were 24 named species concepts that were all seemingly conflict with one another based on the criteria that we use to define a species. And so for this study, I will be using the retrospective reproductive community concept, abbreviated RRCC. That an extant species is a reproductive community of the recent past, extending to the present, within which there was sufficient interbreeding or other cohesive processes to maintain coherence in genealogy and traits, and outside of which there is insufficient or broader coherence. And there are a couple important aspects to this definition. The first of which being that species are historical lineages extending from the past, as in the phylogenetic species concept. And also that these reproductive communities need to be held together by cohesive forces and separated from others with isolation or through isolation forces, as in the biological species concept. And so the RRCC is a sort of in a natural development of these two concepts, and it's also sort of reflective of how systematists have sort of been treating species in their studies sort of practically. One thing I will also note is that the RRCC is an open concept. It is intentionally incomplete in that it doesn't have a set list of criteria that you need to sort of check off to say that it is a species. Something that you sort of need to determine at the lineage level on sort of a case-by-case basis. And so what is species delimitation then? Quite simply, it is the process and methods for inferring species boundaries. And there are a couple important steps in this whole process. First of which is actually identifying reproductive communities that are held together by cohesive forces, separated from others with isolation. And then once you've identified those, you need to make a decision as which of these communities should you designate the rank species. And since the RRCC has no set criteria, no like list that you can go down and sort of check things off, you require evidence, which could take the form of multiple lines of congruent evidence that sort of agree with one another, or it could be an overwhelmingly convincing source of evidence, whatever that should be, to claim the existence that there are species boundaries. And so what are the common forms of evidence that we see in species delimitation studies? The standard, or the most common, is geography and morphological variation. Since the beginning of when people were sort of telling species apart from one another, where these things are distributed and how they appear is sort of the first thing that you really look at. In the past several decades, we've seen the advent of various technologies allowing us to look at molecular data, particularly genes. And with these, we can create gene trees, whether they are individual for seeing how an individual gene might tell us about the relationships, or you can create sort of consensus trees of sort of summarizing all of these together. We've also seen a lot of some multi-species coalescent methods being used. Multi-species coalescent is another method of taking many genes and sort of inferring species boundaries based on them. It's used a lot because it accommodates for discordant gene trees, so gene trees that disagree with one another, along with things like horizontal gene transfer and gene flow between species, incomplete lineage sorting, things like that. And we've also seen, especially in the past 10 years, some things called ultra-conserved elements. They're kind of a popular kid on the block, so to speak, because of the fact that they have been shown to be so useful in species delimitation studies. They're so useful because of the fact that they have this core UCE region, as you can see from this horizontal black line, where when they show up, this core region does not change nearly at all across sort of distantly related. But as you move farther sort of base by base away from this core region, the flanking regions get higher and higher degrees of variability sort of at the base level. And so with this, you can take UCEs from like one species and then another species, align them using the core UCE region, and then compare them using the flanking regions. And it's been shown to be very, very useful for species delimitation studies for that reason. And so what are some more novel methods, stuff that hasn't really gotten as much research to the same degree? And I'm going to focus primarily on repetitive elements, as David mentioned in the sort of lead up to this talk. And those can sort of take the form of anything that sort of is highly replicated in the genome, such as ribosomal DNA. The ribosomal cistron sort of exists as these monomers shown in like purpley blue here. They can go end to end, sort of replicated many times. We may see them as transposable elements, sort of sections of DNA that can copy and paste themselves from one part of the genome to another using an RNA or protein intermediate. Or we can see them sort of like as satellite DNA, which are another sort of tandem repeat where it has monomers stacked end to end, hundreds to thousands of repeats. And they focus or are centralized mostly on telomeres and centromeres, sort of the ends and the centers of chromosomes. And part of the reason why repetitive elements are so interesting as a data source is because they are so abundant. In humans, for instance, transposable elements make up 50% or about 50% of the whole human genome. In some plant species like maize, that can be up to 70% of the whole genome taken up by transposable elements. And so there's a huge amount of data here that we could be using for species delimitation research. And so this is sort of the basis for choosing repetitive elements for the novel methods. When we're analyzing repetitive element data, one thing to keep in mind is that copy number is very important. These are repeated sections of DNA, though the degree to which they are repeated is not the same from species to species or population to population. And so the variation in copy number of these repetitive elements is something that we work on. I'm going to get down into the weeds of methods for a little bit, but we're going to come back out. But the way that we're able to see this is by taking a sort of low coverage whole genome sequence and taking subsamples from that sequence tens of millions of times, these sort of really short subsamples from the genome that we can then map to a particular repetitive element. In this case, I'm using an example of the cistron. And so you've got this repetitive element reference that wherever the subsamples from the genome contains a part of that reference, it can sort of map onto that reference and it starts to pile up. And we eventually get this down here, which I'll refer to as a CNV profile, where we get to see not only the overall copy number of the element, but if there's a specific section of that repetitive element that is more highly replicated. For instance, this red peak down here, how it's sort of in the middle of 28S, we know that that region is more highly replicated than the rest of the ribosomal cistron in this specimen. We can take these CNV profiles and sort of compare them across samples. So we can see here, first 3 samples within a group are all similar and sort of distinct in terms of shape from the other 2 groups. And so we can use these copy number variant profiles, these CNV profiles, as points of comparison to sort of infer species relationships. In addition to copy number, we can also take a look at sequence level differences in the repetitive element. So for instance, if there's a population where the monomer of a satellite DNA is AATTCC, but in another population, it's AATTGC instead, that's a difference in the sequence level of that repetitive element. And that's something that we can pull out from these CNV profiles as well, with sort of this enhanced variant profile here, and then turn it into sort of a sequence of characters that we can compare between samples, similar to how you would with a gene, for instance. And with that, we can do phylogenetic analysis. We can compare them, infer relationships between these samples that we've taken. And that's something that we'll see a little bit more of later in the presentation. Okay, so went over various methods there, but just as a summary, some of the stuff that I'm considering standard for this presentation is geography, molecular variation, molecular data, especially genes and the various things that you can do with those, and ultra-conserved elements. Some more novel stuff that we've not yet seen a whole lot of research on is sort of repetitive elements in the various analyses that you can work with those. One other thing that I will point out is that UCEs and REs have been shown to evolve more rapidly than more conserved parts of the genome. At least in the UCEs, it would be the flanking regions that evolved a bit more rapidly. And part of the reason for this is because REs and UCEs are largely non-coding in that sort of changes in the sequence, mutations, duplications, stuff like that may have less of a fitness consequence immediately. And so we get to see sort of a higher resolution of sort of things happening in these two sources of data. Something that we'll see in the results. So back out of genome world, back into the animals that I'm working with. I work with the genus Bembidian, very large genus, over 1,300 species worldwide. They're real small guys, 1 to 4 millimeters in length generally. They're predatory in nature, live on the edges of bodies of water, so edges of snowmelts, sort of the gravel and sandy shores of rivers, creeks, marshy areas, stuff like that. And it is numerous in genera or subgenera and species groups. And one thing that's important is that many of these are still unresolved in that we don't know the exact number of true species that exist in this genus, and a lot of them have not really yet been studied with species delimitation methods. And so, good thing to sort of pick to test these out. I'll have 2 study groups, the first of which I will refer to as the Recticoli study group, are the Hermopetaphus subgenus, and currently in the literature there are 2 species, Bembidian recticoli and Bembidian avidum. Also in the current literature, there are 2 additional names that will play a part in the story, B. quadrulum and B. provectum. Those are E and F on the chart up here. The current literature says that they are synonymous and that they should be considered the same species. They should not be considered separate. And so a lot of the time, B. provectum is not actually a name that's used when referring to these groups of beings. They're also not a part of the B. recticoli group in the current literature. One thing, the issue though, is something you may be able to tell by looking at these 6 beetles compared with each other, is they look quite similar. There's not a whole lot of morphological differences that are sort of very apparent. And more so than that, with initial sort of genetic data that we sequenced from them, the actual boundaries in the species are not clear. We don't get a good, clear story about how many species there are, where the species boundaries are. And so we know what the literature tells us, but the morphology and the genetic data, it's still cryptic, it's still ambiguous. And so this will be the first of our 2 cryptic species groups that will be running some species delimitation methods on. And then here are various symbols and colors. I'll try to keep consistent to sort of signify the different regions that these little beings are from. The second of the 2 study groups is the Bembidian transversale study group. It has many similar issues as the recticoli group in that there are morphological characters that you can use to tell them apart, but they're not super obvious. And more importantly, the genetic data that we sort of already had on them didn't tell us a clear story about the number of species, where the species boundaries are, and all of that. And so they are a part of the Nearctic clade of the Oxydromus complex. And in the current literature, there are 5 species: Bembidian perspicuum, Sarpedon, transversale, erosum, the last 3 being the 3 that are of interest for this study. This group recently underwent another species delimitation study back in 2020, but the study that happened then was the sort of determination based on morphological differences, and this group has not yet been undergone species delimitation with more involved, especially newer, more novel genomic methods. So this will be the second of our 2 groups that I'll be using as an example. And here's sort of the distribution of the next-gen sequences that we took from the various little creatures. And so, what is the actual experimental design that I'm working with? Starting off with taxon sampling. For the Recticoli group, I sampled from the broader subgenus that it's a part of, along with some outgroup samples. And from those, there were 68 that we took a 4-gene data set from, and 28 of those 68 we sent for next-gen sequencing. And then of that group, it was 39 and 21 that were a part of our core V. Recticoli study group. For our transversali group, since we're off of a previous study, we had a large data set of that sort of 4-gene data set that we were working with originally, 152. But since the original study did not do whole genome sequencing, the data set that we have for that includes 20 samples. And the places that we tried to sample from, we attempted to have different species sampled from geographically close locations. So we know that differences in genes are not just because of geographic proximity. So since this experiment is based primarily on the methods, I'll be sort of laying it out in a flowchart way and then sort of referring back to it as we work our way through. So geographic distribution, context for how we might interpret relationships. I'm going to take a look at some morphological variation within the groups, which could include microsculpture, which is sort of the texture on the surface of the shell of the beetle itself. Going to take a look at differences in male genitalia, something that has long been shown to be a key characteristic for telling species apart from one another, especially in ground beetles. And also taking a look at external body shape. And with each of these, obviously taking a look at them visually, but particularly for external body shape, taking measurements and then doing statistical analysis on the differences in body shape to see if there's a significant difference that we can see there. We've got molecular data, which includes a 4-gene Sanger sequencing data set of 2 mitochondrial genes, 28S and CO1BC, and 2 nuclear genes, topoisomerase and PAD4. With those, we can construct maximum likelihood gene trees, as well as sort of consensus trees from those, and also plug them into a multi-species coalescent analysis. And we've got 2 specific ones that we'll be working with, Stacy and Delineate. We've also got whole genome sequencing of some of the samples. And with those, we can pull out UCE loci, build trees from those. And lastly, we've got repetitive element sequences, which includes the more relatively shorter repetitive elements like transposable elements and satellite DNAs, and the ribosomal cistron and sort of the genes that are included within. And with those, we can build copy number variant profiles, as well as pull out sort of the variant sequences from those repetitive elements and build trees from those. And then there is one last method that utilizes correlation-based data that I'm going to put a pin in and come back to when we get to it. Okay, so what are the aims of this project? The first of which is taking a look at the Recticoli group, testing older and newer methods, and reevaluating the current understanding of theories that we see in the literature. Aim 2 is to take a look at the transversali group, specifically with the newer, more novel methods, and compare those inferences that we see with the more recent literature back in 2020. And then lastly, I want to take a look at the methods themselves and see how they compare for species delimitation inferences in relatively shallow sort of groups. So these groups are, it's very likely that they diverged evolutionarily relatively recently. And so that presents sort of a unique challenge for sort of parsing out the differences with genomic data. And so let's get to the results. So the morphology of the transversali group was something that documented back in the original study, but I'm going to cover it briefly here just to familiarize you. Each column here represents an individual species, and each row is sort of a character that was used to tell them apart. Row A being differences in the shape of the mentum. So this is sort of a mouth part on the underside of the beetle. Row B showing differences in color of the first couple antennal segments. And then C through F being slight but consistent differences in male genitalic structure, whether that is sort of external structures or the more softer structures on the inside. For the reticular group, extensive morphological analysis hadn't really been done in this group at this point. And so I figured where else to start by taking measurements. And so for the measurements, I took maximum width of the pronotum, minimum width of the pronotum, length of the pronotum, maximum width of the eyes, maximum width of the elytra, and then the length of the elytra. And then ran some ANOVA tests on these various measurements and found that these two in particular are ones that are important for telling regions apart. And the way that they are important is not the measurements themselves, but the difference between them. And so I built a sort of box and whisker plot here showing the difference between the maximum and the minimum width, basically how curved inward the pronotum is sort of trying to capture the shape. And what we can see is that if we're comparing the two extremes, rectically from its northern sort of regions, so Alberta, British Columbia, Idaho, Montana, Eastern Oregon, we see a much smaller difference between the maximum and the minimum. Their pronotum is much more sort of square-like, and so we don't see that curve inwards. And then on the other end of the extreme, we've got the rectum down in Arizona, down in southern New Mexico, and of the specimens that were sampled from these localities, they have a significantly different measurement here. It's much more curved inwards and towards the base of the abdomen. And this is a statistically significant difference between rectum and all the rest of recticholy. And so this is the first of a couple times that we'll see rectum sort of separating itself morphologically from the rest of. Taking a look at microsculpture, we've got some photographs here that were taken from a camera mounted onto one of our compound scopes. And so again, going from one extreme to the other, we have our western recticholy along sort of the western coast. It has sort of these darker and hence deeper sort of grooves and sort of geometric shapes on the surface of the shell itself. And these become much more shallow as we move farther inland and farther south until eventually we get to the rectum down here, again, the New Mexican or southern New Mexican region, where the microsculpture is entirely absent. This sort of texture on the surface of the shell itself is not present. And the way that this manifests when you're just sort of looking at the beetle itself is that rectum specimens appear much more shiny. They appear to like reflect a lot more light compared to ones from the sort of western coast. They appear much. And so another case where the rectum sort of separates itself morphologically from the rest of the recticholy. And then lastly, let's take a look at some genitalia. So here are some photographs. We dissected out the male genitalia from various beetles, suspended them in Uparol for microscope slides, and then took photographs of them under the microscope. And what I've done here is sort of taking various photos of various different genitalia, made them transparent, and stacked them on top of each other to sort of get a summary of how shapes might differ. And again, comparing the two extremes, we have B recticholy western and B rectum. And this is probably the most slight out of all of the morphology that we've seen, the most smallest difference. But it is still worth noting that on one end of the extreme is the rectum, the one that sort of keeps standing out with the other sorts of morphology. And what we see is that this sort of dorsal apex here in the rectum is much thinner. It sort of curves down a little bit more. It has a bit of a hooked shape to it. And again, the differences here are very slight, but it is interesting that the two extremes that we see are one of them is rectum and the other is sort of another locality. Okay, got through morphological variation. How about we take a look at some molecular data, especially of the 4-gene variety. So there's a lot to take in here, but what I want you to sort of take away is that the different regions that we're looking at are represented in different colors on the tree, and that also the trees don't tell similar relationships, and that all of the colors are sort of interspersed throughout. We don't get clear, clean clades about telling us sort of the exact relationships that we might infer here. And this is the case for all of the genes that we sequenced. So not just these two, but these two as well. Not really telling us a prevailing clear story, at least when looking at them visually. And this is the case not just in the Recticoly group, but the Transversale group as well. They both have this issue of specimens not separating out into distinct clades. And so interpretation of these trees is difficult. And this is the case for all of the genes. And so the genes by themselves maybe don't tell us a clear story. So what if we sort of compile them all together, have a method that we can have maybe there's a prevailing pattern that we're not seeing. And so moving on to some multi-species coalescent analyses. The first of the two that we were working with was an algorithm called Stacy. And in the Recticoly group, the inference that is made is not much better than the individual genes. Stacy sort of infers that all of Recticoly is one distinct species, not to be separated out any further than that, which, you know, may be the case, but I feel further investigation is probably needed. And then the Transversale group as well, it has the same issue of not separating out into any further resolution than what we see here. What about the other one, though? So we take a look at delineate, which is our second of the two multi-species coalescent methods. And if we zoom in here, we can see that there's a little bit more resolution in the tree. At least, if not with the at a species level, we get to see at the very least population level, there is certain clades that are beginning to form, and they are at least somewhat consistent with sort of the geography, the different regions that we were pointing out previously. One thing I will point out is this first word at the tip of each of these, Humboldtensi. Humboldtensi is another species within the same gene or subgenus. And so what delineate was doing here is it was able to separate out the population groups, but when asked what species all of this was, it was like, hey, it's just Humboldtensi. It's not its own species, and so you should just consider it Humboldtensi, which is inconsistent with the morphology that we see along with geographic distribution and other evidence. And so we'll take it how it is for now. Okay, so we move past the 4-gene synchronous data. What do we learn from our whole gene data sets and the data that we got? Starting with UCE analysis. And this is the first time that we see distinct clades that correlate with geographic distribution. And this was amazing to see. It was very exciting because at this point in my research, I thought that all hope was lost, that there was no solution, that I should just hang my coat up and walk into the ocean. Like it was rough, but we got somewhere with it. And what we see is that quadrulum is a separate clade on its own, apart from the broader clade that contains the Recticoly group. And that Rectum is a sister clade to the rest of Recticoly, which is sort of interesting and may be consistent with some of the morphology that we were seeing. For the Transversale group, these are the results. Chorogenoma groups as its own clade, but we start to see issues here between Erosum and Transversale. they don't quite have the same clear story as the Recticoli group did, or sort of core genoma separate from everything else. And so perhaps we need to take a look at some other data sources, see what they tell us. And so we take a look at the repetitive element sort of variance that we can put into a tree. And again, we see distinct clades, which are consistent with the geographic distribution, which is also really cool to see. And another thing that's very cool to see here is it provides a similar amount of resolution as ultra-conserved elements, sort of like the popular kid on the block right now. And so this bodes very well for repetitive elements as a source of data for species delimitation study. And also the number of repetitive element sequences that went into this was, there were 1,000 sequences that we used to generate this tree, which is, to my knowledge, the largest analysis using that sort of data of this kind thus far, which is just a fun note. And so for the transversale group, we take a look here, same thing of the proteoma separating into its own clade, and then the relationships between transversale and erosum still being intermixed, still being ambiguous, and so no real conclusions about where the boundaries are between those two being drawn yet. Okay, so I just showed you a lot of previews. I'm gonna sort of try and summarize it all into some prevailing patterns that we see. So here's a summary table that I constructed. The rows being the individual specimens that we took and sequencing from, and the columns being the results of the various analyses that we were looking at. And so the patterns I want to point out is that B quadrilum down here in the green, in every single method, separated itself as something different from Recticoli as a whole. And also that these two columns over here, UCE and RE variant profiles, are the two sort of high resolution methods that we see. It sort of separates things out to the highest degree of detail. And then in those two high resolution methods, we see B porectum as being claimed as separate from the rest of Recticoli, which is sort of consistent with what we saw from the morphology. And this map over here is just showing where those highest resolution populations would be if you plotted them onto a map, just to show that sort of the different groupings that were inferred are sort of geographically sound, that they, could have feasibly happened. And another summary table for transversale, we see that core genoma is sort of maintained as a separate entity outside of the other two, and that these two, erosum and transversale, are, the relationships are ambiguous and cryptic still. Okay, we got through all of that. And so what are the last little straggling methods that we'll have to talk about related to repetitive elements? And so we can just take a look at the CNV profiles themselves. And so this here is various different ribosomal DNA sequences compared between the different regions of Recticoli. And the only thing that I really want to point out here is that if you sort of take a look at them visually, that the middle quadrilum down here, it seems that there is a different proportion of coverage across the whole ribosomal cistron as compared to Recticoli as a whole. Just sort of the overall shape, the depth is much lower compared to these top four samples. And then with transversale, if we do the same thing with them, we see a slight difference in proportion of coverage between transversale and erosum, which is nice to see. We also see quite a bit of variability within core genoma. And so these are multiple profiles that have sort of been stacked together and sort of scaled so that each individual profile is sort of this light gray, and it gets darker to sort of the darker gray. And so core genoma here showing that it has quite a bit of variability in at least its ribosomal sequence. When we were also taking a look at repetitive elements, we were extracting them in silico from our whole genome sequences, and we got thousands and thousands of these things back. And we were sort of looking through them and just sort of visually telling the differences, just to see if there's any patterns that we could tell. And some of the cases ended up being that some of the repetitive elements show similarities within given groups and differences with other groups, sort of consistent patterns that we might be able to interpret. The vast majority of the time, a lot of the repetitive element sort of CNV profiles ended up looking like this, where there's not a whole lot of prevailing pattern here that we can interpret super well. And this was the vast majority of the repetitive elements that we saw. This is not super surprising because of the fact that most repetitive elements are not actively replicating themselves. Many of them are dormant, sort of essentially fossilized or like fossils of DNA of events long past. And so sort of the patterns that we'd see here are not super relevant for asking this more short-term, sort of shallow evolutionary question. But still, we've got thousands of them. There must be something that we can do with all of this data. So this is where I get to the thing that I put a little pin in earlier. With the program that we have to create CNV profiles, we also have the ability to get correlation data from them. And we get correlation of specimens within group. And so if you designate a certain group, all the profiles in here will have a correlation value associated with that, and then correlation between groups. And so this given sample compared to, say, this given sample down here. And the greater the difference between these two correlation values might suggest that there is divergence happening, at least with how these repetitive elements are distributed throughout a genome. So we wanted something to capture this difference in correlation values. So we set up some hypothesis testing methods. For the Recticoli group, we grouped the various specimens into a couple of different hypotheses. First of which being groupings based on the phylogeny of the UCE data. Second grouping being based on distinct external morphology. Third grouping being based on the current literature that the Recticoli group is a group on its own, and that co-rectum and quadrulum are lumped together as something separate. And then the last hypothesis being based on the phylogeny that we saw from the 4-gene data, that all of Recticoli, including co-rectum, is one thing, and that quadrulum is something separate entirely. And so we did some analyses, and these are the results that we got. So the numbers within each of the cells, within each of the groupings, represents the number of CNV profiles that met a certain threshold of difference in those two correlation values that I was referring to. And so the higher the number, the more CNV profiles that met that threshold of difference. And so as sort of averages from each of the hypotheses, we see that the UCE phylogeny and that the 4-gene phylogeny has the highest overall averages among these hypotheses that we looked at. I'll interpret those in a moment. The Transversali group, it was much simpler. There was only really one question that we were asking, is if there are 3 species in this group or there are 2 species in this group, because of the sort of confusing results that we got from the phylogenetic trees between Orosum and Transversali, sort of the pink and the green in those trees. And here are the results from that analysis. The 3 species hypothesis showing a greater average number of CNV profiles that met that threshold that we designated. So there's a bit of a problem with this method that I just presented using correlation data, in that with correlation-based statistics, bin size plays a big factor, or rather sample size, where smaller bins seem to show greater within-group correlation. So if you've got a sample of 2, it's more likely that those 2 samples will have more in common with each other than if you have a sample of 10. And if there's any individual within that 10 that is even slightly different, it could bring that correlation value down. And so what we end up seeing is as bin size grows, for the hypotheses that we're looking at, the number of profiles that meet that threshold we were looking at decreases. And so how much is this impacting our data? Where do our observed values fall along here? So this is some hypothetical data. Even if there was a skew based on bin size, we would still expect our observed values to sort of rise to the top. So be return more CV values than just randomly assigned to these bins. And so what do we actually see? We saw that our observed values for hypothesis 1 or rectified group falling pretty much in line with the randomized distribution, which, so what does that tell us about this? First off, it tells us that if we're going to use this method in the future, we need to normalize for bin size. We also need to include a statistic value so that we know even if we saw this sort of pattern here, we would need some sort of statistic like a p-value to tell us if it is significantly different from what is random. And then also further testing is needed with this method. The groups that we're using to study this and sort of try out this method for the very first time are cryptic and ambiguous with their relationships to begin with. And so additional sort of study groups to sort of troubleshoot this whole thing would be necessary. But it was a cool idea, and I look forward to seeing how this method may develop. So let's talk about conclusions. For the first of my aims, I wanted to take a look at the rectified group, test older and newer methods, sort of reevaluate the species boundaries that we see in the literature. And I have two major conclusions here. The first of which being that Bimbidium quadrulum and Bimbidium correctum are in fact separate species, which cannot be considered synonymous as they are in the literature. And this is based on sort of that summary table of like those three quadrulum being separate in every single method that we looked at. And Bimbidium correctum is a new species separate from Bimbidium recticoli. This is based on the morphological data that we took a look at, as well as our sort of highest resolution that is separating correctum as being sister to all other recticoli. For aim 2, we wanted to take a look at the transversale group and particularly newer species delimitation methods. And we have two major conclusions for this. That newer species delimitation methods reinforce Bimbidium corgenoma as a separate species. There's nothing that we really found that pulled this into question. It sort of agrees with what is written in the literature. And the relationships between Bimbidium transversale and Bimbidium erosum remain unresolved. The genomic data that we took a look at, in no such case was there a clean separation between the two species. And so further study would probably be needed to resolve that relationship. Okay, so what about the methods as a whole? We were concerned about how they perform in sort of shallow evolutionary groups. So what did we see sort of across the board? We saw that morphological variation is useful. It's a good context for interpreting the other results that we see. It'll always be useful. Sort of morphology is the first thing that we look at most times anyways. For gene data sets, specifically a small gene data set of our 4 here, individual gene trees may not tell you a whole lot within shallow evolutionary divergence. And so it's worth trying out, but you may not get the results you're looking for. With multi-species coalescent analysis, Stacy, when using the same sort of data set, was not able to provide any greater level of resolution. But Delineate, on the other hand, even though it was not able to separate things out at a species level, it did give us greater resolution at a population level. And so we got to see some extra population resolution there, even given the same amount of data as Stacy. And so suggests that Delineate is a bit more robust in its ability to do so. Ultra-conserved element loci and repetitive element variant sequences both performed very well in terms of providing resolution of relationships between populations and species. And so, and it also is very promising that a source from repetitive elements is able to perform to such a degree, sort of to compete, or rather to a similar level as UC loci are. And for our last two, the correlation-based hypothesis method, it's a bit preliminary still. Further testing is needed before we really draw any conclusions about how effective it is. And visual comparison of C and V profiles is good for getting context of how things may be related, but it is very time consuming, and it's sort of just scanning through an ocean of these colorful sort of landscape type plots. And I've certainly spent many, many hours trying to figure out if there is a pattern or not that way. And that's the conclusion for my results. I'd like to take a little moment to thank some people, especially the people that helped me make this become a reality. I would like to thank the Madison Lab, both current and past undergraduate and graduate. A special thanks goes out to June Kim for taking photographs of all of our genitalic preps. A big, big thank you to David Madison for helping me with pretty much everything at one point or another. This genuinely would not exist without your help. Another huge thank you to John Sproul for the hours of meetings that we've done and sort of helping me out with interpreting repetitive elements data. And also a thank you to my committee members, Jonathan Kaplan and Felipe Barreto, for helping me stay on track with all of this, making sure that my boxes are getting checked and sort of helping me out at the institutional level. I would also, of course, like to thank all of the people that may not have been involved in the research itself, but certainly helped me get through grad school. All of the game nights and the watch nights and the happy hours and the meals and D&D, it was all wonderful to get to know everyone here. It really, really did make being in grad school, like, I wouldn't say worthwhile because I'm getting a degree out of this, but it made it enjoyable, certainly, more so than it really otherwise. And of course, thank you to my family. I have a brother here to help me out with moral support. And thank you to my parents, and my other siblings, and my young nephew Leo, and all of the fur babies that I can give cuddles to whenever I go home. I will relieve you of my speaking voice for now. You have questions if we have any time.
So, any questions?
Probably getting results from the UCE data. Maybe a little taking my butt, making sure that I get significant results from this whole thing. Yeah, that was very nice. Yes.
How many individual beetles do you have to use for each method to feel confident that that method was actually showing something consistent and wasn't just an individual difference?
Yeah, that's sort of a hard question, but a lot of the time, for next gen sequencing, so Illumina sequencing, we wanted to have at least 5 samples per sort of region that I talked about. So like Western, Northern, Southern rectum, with a particular emphasis on the rectum aspect of things, because they seemed to visually look different from everything else. And I wanted to make sure that I was capturing sort of the information there. You know, obviously more is better in sort of, you know, a lot of analyses, but that's sort of what we were working with.
Did you see a lot of variation in the morphology, like within each location, or what you consider a species, or, you know, the grouping that you had? Like, let's say you have a replicate of 5, like how much variation is there? And can you use that information, like the coloring? I know you mentioned that, like, is that super consistent within one group, or is it variable?
So in terms of coloring, in some of the photographs that you saw, you might like see or interpret like a slight like green hue or a slight like blue hue. And a lot of that has to do with sort of lighting. And so for that type of coloring, we don't really take that into consideration unless it's like very prominent. But in terms of like at a specific location, seem to be relatively consistent. Of course, there's like sexual dimorphism where males and females might appear more or less shiny than one another. And so for stuff like that, like microsculpture, where shininess is kind of a factor, we use like all male specimens to try and make sure that we're controlling that variable.
So when I've used species limitation methods, like one of the most annoying parts of the whole thing is just wrangling the input files, because it seems like every program requires a different set of files with differences in like what parameters. As you've been exploring all these different, and I'm sure there are some that you didn't even present here over the years that you've tried, like, were there any that you can say like, this one is abused, or like, this one was horrible, no one should use it?
Believing it's pretty hard. It's not really just one program, it's like 3 with several steps in between. In terms of easy to use, I would say that Repeat Profiler, sort of the program that I use to generate a lot of these CNV profiles, was pretty easy to use. It only really required like 2, maybe 3 input files if you want to group things specifically, like value out of it. Yeah, so I would say that that one was probably the easier one to use among them.
Yeah, well, okay, so now we'll transition to the closed door part.